For over a century, the story seemed settled. Vikings reached North America around 1000 AD, stayed briefly at Lands aux Meadows in Newfoundland, then sailed back to Greenland and Iceland, leaving nothing behind but archaeological ruins and fading sagas. The encounter was portrayed as fleeting, hostile, ultimately insignificant. Two worlds touched, then separated forever. But in 2010, a team of geneticists in Iceland made a discovery that shattered this comfortable narrative. Hidden inside the DNA of living Icelanders, passed down through 40 generations of mothers and daughters, they found something that should not have been there. A genetic signature that could only have come from one place: the indigenous peoples of North America. The Vikings had not just visited the New World. They had brought Native American women back across the Atlantic Ocean, and those women had children whose descendants walk the streets of Reykjavik today. The lead researcher was Agnar Helgason from Decode Genetics in Reykjavik, working with colleagues from universities across Iceland and Spain. Their study, published in the American Journal of Physical Anthropology in 2010, began with a straightforward question about Icelandic genetic diversity. They were building a comprehensive database of mitochondrial DNA, the genetic material passed exclusively from mother to child without recombination. Mitochondrial DNA creates clear maternal lineages stretching back through time, making it the perfect tool for tracing ancient population movements. The team analyzed samples from hundreds of Icelanders, categorizing them into haplogroups, the major branches of humanity's genetic family tree, defined by shared mutations indicating descent from common ancestors. Most Icelandic mitochondrial DNA fit the expected patterns. Haplogroups H, J, T, U, and K dominated the population, all characteristic of European ancestry, reflecting the Norwegian and Celtic origins documented in historical records and archaeological evidence. These lineages told the familiar story of Norse settlement, beginning in 874 AD, when Ingólfur Arnarson became Iceland's first permanent settler, followed by waves of Norwegian chieftains, fleeing King Harald Fairhair's consolidation of power, along with Celtic slaves and wives from Norse raids on Ireland and Scotland. But then the geneticists found something completely unexpected. 4 Icelandic families carried haplogroup C1, not C1a or C1d, which appear in Asia, not C1b, which appears across North America from the Great Lakes to the Amazon. Instead, C1e, a sublineage of C1, had never been documented anywhere in the world before this study. Haplogroup C1 is extraordinarily rare in Europe. It appears in less than 0.2% of European populations. Its presence in Iceland demanded explanation. The research team systematically eliminated potential sources. Could it have come from Asian populations? No. The specific sublineage C1e showed no matches in comprehensive Asian genetic databases. Could it represent recent immigration? No. The genealogical records, meticulously maintained in Iceland for over 1,000 years, showed these families descended from lineages established in the early settlement period. Could it be a mutation that arose independently in Iceland? No. The molecular structure clearly placed it within the C1 haplogroup family that evolved in the Americas. Before we go further, I am curious. What country are you watching from, and what time is it where you are right now? Drop it in the comments. I love seeing how far lost evidence reaches across the globe. The geneticists traced the 4 C1 families back through Iceland's exceptional genealogical records, some of the most complete in the world. All 4 lineages converged on ancestors living in Iceland before 1700 AD. 2 of the families traced back to individuals born in the early 1700s in northern Iceland. Another traced to the Vestfjarðir region, the 4th to the Skagafjörður area. But the genealogical records, detailed as they were, could only reach so far into the past. Church records became sparse before 1600. Settlement-era documentation existed primarily in the Land Nemabok, the Book of Settlements, compiled in the 12th century from oral traditions about the original Norse colonists. The key to dating the C1 lineage's arrival came from molecular clock analysis, a technique that estimates when genetic lineages diverged by measuring mutation accumulation rates. Mitochondrial DNA mutates at a relatively consistent pace, allowing geneticists to work backward from present-day genetic diversity to calculate when lineages separated. The team compared the C1e sequences from Iceland with C1 sequences from Native American populations across North and South America. The analysis revealed that C1e had split from other C1 lineages approximately 1,000 years ago, coinciding precisely with the Norse presence in North America, documented archaeologically at Lons á Meadows and described in the Icelandic sagas. The implications were extraordinary. At least one Native American woman, possibly more, had traveled from North America to Iceland during the Viking Age. She had integrated into Icelandic society, and she had children. Those children survived, married, and had their own children, passing her mitochondrial DNA through an unbroken maternal line for 40 generations until it appeared in the genetic testing of her descendants a millennium later. This was not a story of brief contact and departure. This was a story of a permanent genetic legacy. But the discovery immediately raised questions that the genetic evidence alone could not answer. Questions that continue to trouble researchers and indigenous advocates today. How did this Native American woman or women end up in Iceland? The sagas provide disturbing hints, but no definitive answers. The saga of the Greenlanders and Eric the Red's saga both describe encounters between Norse explorers and indigenous peoples they called Skraelings, a term whose meaning remains debated, but was clearly used pejoratively. These accounts describe both trade and violence. In one passage, the Norse trade red cloth for furs. In another, they massacre indigenous people sleeping under kayaks. In yet another, indigenous warriors attack the Norse settlement, forcing the abandonment of the colony. The power dynamics were stark and brutal. The Vikings were not peaceful explorers. They were raiders and slavers across Europe, notorious for capturing people, particularly women and children, during their attacks on Ireland, Scotland, England, and Francia. Thousands of Celtic and Anglo-Saxon slaves were brought to Iceland during the settlement period, documented both in genetic studies showing significant Celtic maternal ancestry and in historical sources describing thralls as fundamental to the Norse economy. The same culture that enslaved people across the North Atlantic had now encountered an entirely new population in North America. The possibility that native women came to Iceland voluntarily seems remote, given both Viking cultural practices and the saga descriptions of violent encounters. The genetic evidence cannot tell us whether the C1 woman came as a slave, a captive, or in some other capacity. The historical record has not preserved. It cannot tell us her name, though she may have been given a Norse name upon arrival. It cannot tell us whether she was taken from Vinland, the region around Lons á Meadows, or from somewhere else along the North Atlantic coast during expeditions the sagas never recorded. It cannot tell us whether she came alone or with others whose lineages failed to survive the genetic bottlenecks of medieval Iceland's harsh conditions, where infant mortality exceeded 50% and periodic famines devastated the population. The origins of this woman remain unknown. The CE1 discovery forced archaeologists and historians to reconsider the scale and duration of Norse activity in North America. Lons á Meadows, discovered in 1960 by Norwegian explorers Helge and Anne Stein Ingstad, remains the only confirmed Viking settlement in the Americas. The site consists of 8 buildings constructed from turf over wooden frames, identical to Norse structures in Greenland and Iceland. Radiocarbon dating places the occupation around 1000 AD, exactly matching the saga chronology. The buildings include 3 dwellings, a forge for working iron, carpentry workshops, and storage structures. This was not a temporary camp. This was a base designed for extended operations. But the site's small size, housing perhaps 70 to 90 people, and its brief occupation period, probably only a few years based on artifact analysis and the minimal accumulation of cultural deposits, had long suggested limited Norse presence in the New World. The genetic evidence challenged that interpretation. If Vikings brought Native American women back to Iceland, the contact must have been substantial enough to establish relationships, whether forced or otherwise, that resulted in women leaving their homelands for the North Atlantic. This implied either longer occupation periods than the archaeology at Lance Meadows suggested, or additional sites yet to be discovered, or repeated voyages over multiple years creating sustained interaction. The sagas describe exactly such repeated voyages. Leif Eriksson's initial exploration was followed by his brother Thorvald, who died in a skirmish with indigenous warriors. Then came Thorfinn Karlsefni's expedition, which brought women and livestock, clearly intending permanent settlement. The saga describes this colony lasting 3 years before conflicts with the Skrælings forced abandonment. Finally, Freydis Eriksdottir led an expedition that ended in violence among the Norse themselves. These were not isolated encounters, but a sustained Norse presence, spanning at least a decade, possibly longer if undocumented voyages occurred. The archaeological evidence from Lance Meadows supports sustained Norse activity, but also reveals its limitations. The site produced Norse artifacts including a bronze pin, a bone needle, iron nails, stone weights used for spinning wool, and waste flakes from iron smelting. These objects confirmed the Norse presence, but also suggested the settlement served primarily as a staging area for further exploration and resource extraction, rather than as a permanent colony. The absence of extensive middens, the trash heaps that accumulate at long-term settlements, indicated relatively brief occupation. The lack of a cemetery suggested no one intended to stay permanently, but Lance Meadows may represent only one node in a larger network. Recent archaeological surveys using satellite imagery and LiDAR technology have identified potential Norse sites further south, though none have been confirmed through excavation. The Point Rosie site in Newfoundland, identified by archaeologist Sarah Parkak using satellite remote sensing, produced features that initially appeared to be Norse, but later analysis proved inconclusive. This demonstrates the difficulty of confirming additional Viking sites without unambiguous artifacts. The Tanfield Valley site on Baffin Island in Arctic Canada has produced Norse artifacts, including bronze fragments and spun yarn. These finds suggest Norse presence far north of Newfoundland, but the dating and context remain debated. The question of where exactly the C1 woman came from, geographically within North America, remains frustratingly unresolved. Haplogroup C1 appears across indigenous populations from Alaska to South America, making it impossible to pinpoint origin based solely on the haplogroup designation. The Norse sagas describe Vinland as having specific environmental characteristics: meadows suitable for cattle, wild grapes, salmon-filled rivers, and timber. Those descriptions have led most scholars to identify Vinland with the region from Newfoundland south to the Gulf of St. Lawrence, possibly extending to New Brunswick or even New England. The indigenous peoples in this region when the Norse arrived would have been ancestors of the Beothuk in Newfoundland, the Mi'kmaq in the Maritimes, and possibly Algonquian groups further south. The Beothuk, tragically, were completely exterminated by European colonization. The last known individual died in 1829, making it impossible to test whether they carried C1 haplogroup lineages related to the Icelandic C1e. The Mi'kmaq and other maritime indigenous groups do carry C1 lineages, but no comprehensive genetic surveys have been conducted specifically comparing these populations to the Icelandic C1e to determine potential closer relationships. This represents a significant gap in the research. A detailed comparison of the Icelandic C1e with C1 lineages from Mi'kmaq, Maliseet, and other northeastern indigenous groups could potentially narrow the geographical origin, but such a study requires collaboration with indigenous communities who have legitimate concerns about genetic research, given its history of exploitation and misuse. The fate of the C1 woman in Iceland remains similarly mysterious. Did she survive the voyage? The North Atlantic crossing from Newfoundland to Greenland to Iceland covered roughly 3,000 kilometers across some of the world's most dangerous waters. Norse Nars, the cargo vessels used for these journeys, were seaworthy but offered minimal shelter. Crossings could take weeks if winds were unfavorable. Starvation, dehydration, hypothermia, and disease killed many passengers on such voyages. But the genetic evidence proves at least one. C1 woman not only survived the journey, but lived long enough in Iceland to have children who themselves survived to reproductive age. What was her life like in Iceland? The Norse brought thousands of Celtic slaves to Iceland during the settlement period, particularly women who were integrated into households as servants, concubines, and occasionally wives. Genetic studies show approximately 60% of Icelandic mitochondrial DNA derives from Celtic sources, compared to only about 25% of Y chromosome DNA, indicating Norse men systematically took Celtic women. The same pattern would have applied to the C1 woman. She would have been exotic, foreign, probably unable to speak Norse initially, completely isolated from her culture and people, living in a harsh subarctic environment utterly unlike her North American homeland. The archaeological record of Norse Iceland reveals brutal social stratification. Wealthy chieftains controlled vast farmsteads with numerous slaves. Living conditions for thralls were harsh, sleeping in the coldest, darkest parts of longhouses, eating inferior food, working exhausting hours. Female slaves faced sexual exploitation as a routine aspect of their servitude. Children born to slave women inherited slave status unless the father chose to acknowledge and free them. The C1 woman's children must have been acknowledged in some capacity, since they survived and their lineage persisted, but the circumstances remain unknowable. Did their father free them? Did he die, leaving them in uncertain status? Did they escape slavery through some other means? Iceland's exceptional genealogical records, which should in principle allow tracing the C1 lineages back to specific individuals, become frustratingly vague in the early settlement period. The land Namabok lists approximately 400 primary settlers and their families, but it focuses on prominent chieftains and largely ignores slaves and lower status individuals. Women appear primarily as wives and daughters of important men. Slaves receive mention mainly when they cause problems or when their descendants later achieve prominence. The C1 woman, if she were a slave, would likely never have been recorded by name. This absence from the written record reflects the broader erasure of indigenous peoples from Viking Age narratives. The sagas treat the Skrælings as dangerous obstacles or sources of trade goods, not as peoples with their own complex societies, histories, and cultures. Modern archaeology has begun to correct this imbalance, revealing the sophisticated indigenous societies the Norse encountered. The Dorset culture occupied Newfoundland when the Norse arrived, having developed specialized technologies for hunting seals and whales in Arctic waters. Further south, late woodland peoples practiced agriculture, built substantial villages, and maintained extensive trade networks. These were not primitive societies, but complex cultures with deep knowledge of their environments. The power imbalance between Norse and indigenous peoples, while severe, was not absolute. The sagas describe indigenous military victories that forced Norse abandonment of their settlements. The indigenous peoples possess superior knowledge of local geography, resources, and conditions. They outnumbered the Norse settlers by vast margins. The genetic evidence of at least one Native American woman in Iceland proves contact occurred, but the absence of a more widespread genetic legacy suggests this contact remained limited. If the Norse had established permanent colonies and sustained peaceful relations, we would expect to find the C1 haplogroup distributed more widely in the Icelandic population, rather than appearing in just 4 families. The scientific community's reception of the C1 discovery was initially cautious, then increasingly accepting as the molecular evidence withstood scrutiny. Some researchers proposed alternative explanations. Could the C1 lineage have entered Iceland through Inuit populations? No, because the specific C1e sublineage did not match Inuit genetic profiles, and Inuit contact with Iceland occurred much later, in the 18th century, too recent to explain the genealogical depth of the Icelandic C1 families. Could it represent ancient Asian migration to Europe through routes other than North America? No, because the molecular structure clearly placed it within American C1 evolution rather than Asian C1 evolution. Follow-up studies have attempted to find additional C1 lineages in Iceland through expanded sampling, but to date, the original 4 families remain the only confirmed cases. This rarity suggests either that very few Native American women came to Iceland, or that most such lineages went extinct through the demographic accidents that eliminate many genetic lines over time. Medieval Iceland's population fluctuated between 30,000 and 50,000 people, experiencing periodic bottlenecks from volcanic eruptions, famines, and the Black Death in the 14th century, which killed approximately half the population. Small genetic lineages could easily disappear through random chance during such catastrophes. The C1 discovery has implications beyond simply proving Norse indigenous contact. It demonstrates that the Vikings' North American ventures, while ultimately unsuccessful as permanent colonies, were substantial enough to result in transatlantic movement of people. This challenges the minimalist interpretation of Norse America as a brief episode of exploration and retreat. It suggests the Norse invested significant effort in attempting to exploit North American resources, particularly timber, which was desperately scarce in Greenland and Iceland. The sagas describe ships loaded with valuable timber from Vinland being brought back to Greenland, where wood was worth its weight in silver. The genetic evidence also raises ethical questions about how we remember and represent Viking Age exploration. Popular culture often romanticizes the Vikings as bold explorers discovering new lands. The DNA evidence forces acknowledgement of darker realities. The Native American woman whose mitochondrial DNA persists in Iceland was almost certainly brought there against her will, enslaved, separated forever from her people and homeland. Her presence in the genetic record testifies not to adventure and discovery, but to violence, captivity, and cultural destruction. Yet she survived. She had children. Her lineage persisted through a millennium of Icelandic history. She became, in a sense, one of Iceland's founding mothers, though the society that erased her name would never have acknowledged her as such. Indigenous communities in North America have had varied responses to the genetic findings. Some see it as confirmation of what oral traditions already indicated, that their ancestors encountered Europeans long before Columbus. Some view it as further evidence of the violence European contact has consistently brought to indigenous peoples. Some express frustration that their genetic material is being studied and discussed by outside researchers without their full participation or consent. The ethical frameworks for conducting genetic research with indigenous communities have evolved significantly since 2010, with greater emphasis on community consultation, shared decision-making about research directions, and recognition of indigenous data sovereignty. The question of whether additional Native American women came to Iceland beyond the documented C1 lineage remains open. Comprehensive genetic surveys of the entire Icelandic population could potentially identify other American lineages. Such surveys would need to test thousands of individuals to ensure adequate coverage, and they would require sophisticated analysis to distinguish ancient indigenous American ancestry from later admixture after European colonization of the Americas. The cost and complexity of such a project has prevented its implementation, but advancing genetic technologies and decreasing costs may make it feasible in the coming years. Similarly, systematic genetic surveys of indigenous communities in northeastern North America, conducted with full community participation and consent, could potentially identify populations sharing closer genetic relationships to the Icelandic C1e lineage. This could narrow the geographical origin from the broad region of northeastern North America to specific territories. Such information would be culturally significant for the indigenous communities involved, confirming their specific ancestors' role in this early transatlantic contact. But it would require building trust between indigenous communities and researchers, a process complicated by the long history of extractive research that has taken indigenous biological materials, knowledge, and cultural information without proper recognition or benefit to the communities themselves. The Icelandic C1 woman represents one of the great mysteries of Viking Age history. We know she existed. We know she came from North America. We know she survived the journey to Iceland. We know she had children, but everything else, her name, her homeland, her people, the circumstances of her capture, her experiences in Iceland, her thoughts and feelings, all of it remains locked behind a millennium of silence. The genetic evidence gives her a presence, a reality that cannot be denied or erased, but it cannot give her a voice. It cannot restore what was taken from her. It can only confirm that she was there, that her story, however invisible in the written record, became permanently woven into the genetic fabric of Iceland. The broader implications for understanding Norse exploration continue to unfold as new evidence emerges. Recent discoveries of Norse artifacts in Canadian Arctic sites, previously thought to be purely indigenous, suggest Norse trade networks extended much further than the sagas recorded. Neutron activation analysis of whetstones from Arctic Canada sites matches the geochemical signature of Norwegian schist, proving these objects were transported from Scandinavia across the Atlantic and then traded northward through indigenous networks. This implies sustained contact and exchange rather than isolated encounters. Each new finding adds complexity to the picture, revealing that the Norse North American ventures, while failing to establish permanent colonies, created a web of connections whose full extent we are still discovering. The C1 discovery also demonstrates the power of genetic archaeology to reveal hidden histories that left no written record. How many other ghost lineages exist in human populations, testifying to encounters and migrations that never made it into historical documents? The genetic record preserves information that survives when documents burn, when oral traditions fade, when archaeological sites erode away. It creates a parallel archive of human history, written not in ink, but in the molecular structure of DNA, passed down through generations regardless of whether anyone remembered or recorded the stories those sequences encode. For Iceland, the C1 lineage adds another layer to the complex genetic heritage of a population already known to be a mixture of Norse and Celtic ancestries. Icelanders carry the genetic legacy of Norwegian chieftains, Irish and Scottish slaves, and now, we know, at least one woman from the indigenous peoples of North America. This diversity contradicts simplistic narratives of ethnic purity that have sometimes been projected onto Iceland, instead revealing a founding population drawn from across the Viking world and remarkably even beyond it. The C1 woman, whoever she was, became part of Iceland's foundation, her genes spreading through 40 generations of descendants who carry forward a fragment of North American indigenous heritage in the far North Atlantic. The unanswered questions remain. How many other Native American women came to Iceland? Did any men come as well? And if so, Why do we find no trace of them in genetic studies? This could be explained by the nature of the Y chromosome inheritance, which requires unbroken male lines to persist, versus mitochondrial DNA, which only requires unbroken female lines. Male captives might have been killed, might have died without children, or might have had only daughters, eliminating their Y chromosomes from the Icelandic gene pool, while allowing their autosomal DNA to persist undetected. But some researchers and genetic genealogists have raised another intriguing possibility. Could the genetic flow have gone both directions? The C1 evidence proves Native American women ended up in Iceland. Some have claimed to find evidence of Norse paternal lineages in northeastern Native American tribes, specifically haplogroups R1a and R1b appearing in Mi'kmaq, Penobscot, and Passamaquoddy populations. These are European Y chromosome haplogroups that would indicate Norse fathers and indigenous mothers producing children who remained in North America rather than traveling to Iceland. However, this claim remains highly controversial and unverified through peer-reviewed genetic studies. The presence of European haplogroups in native populations could equally result from post-Columbus contact, from later European fishermen and traders, or from French colonial period admixture, making it extremely difficult to distinguish potential Viking-age genetic contribution from later European colonization. Without ancient DNA samples from pre-Columbian indigenous remains in the contact zones showing these haplogroups, or comprehensive genetic surveys specifically designed to date when these lineages entered native populations using molecular clock analysis, the R1 claim remains speculation rather than established fact. It represents an intriguing research question, not a confirmed discovery. Were there other Norse sites in North America beyond Lons Meadows where longer-term contact allowed for relationships and family formation? Did the C1 woman come from a single expedition, or did multiple voyages bring indigenous peoples to Iceland over several decades? What happened to the indigenous perspective on these encounters? The Norse sagas provide the European viewpoint, but we have no indigenous written records from this period to show how the Beothuk, Mi'kmaq, or other northeastern peoples understood these strange visitors from across the ocean. Oral traditions may have preserved memories of these encounters, but connecting those traditions to specific events a millennium ago presents enormous uncertainty. Some Mi'kmaq traditions speak of ancient visitors from across the sea, and some researchers have noted what they interpret as Indo-European themes appearing in Mi'kmaq, Penobscot, and Passamaquoddy folklore. However, similar themes appear in many unrelated cultures worldwide, and cultural diffusion through later contact provides equally plausible explanations. The indigenous perspective remains essential, and understanding it will require careful, collaborative research that respects community knowledge and concerns. Without clear linguistic or archaeological corroboration, folklore analysis alone cannot establish Norse contact, though it adds another layer to the puzzle of what indigenous oral traditions might preserve about these encounters. The climate of Viking-age North America may have facilitated longer occupation than the archaeology currently shows. The medieval warm period, lasting from approximately 900 to 1300 AD, created more favorable conditions in the North Atlantic and northeastern North America. Reduced sea ice made navigation easier. Milder temperatures in Newfoundland and Labrador would have made the environment more hospitable to Norse farming practices. But this warmth was temporary. As the climate cooled into the Little Ice Age, beginning around 1300, the already marginal Greenland Norse colonies struggled and ultimately failed. The North American ventures, even more distant and difficult to supply, would have become completely untenable. The Norse abandoned Greenland entirely by the 15th century, just as Columbus was preparing his voyages from Spain. The genetic legacy outlasted the physical presence. Long after the last Norse ship sailed from North American shores, long after Lance Meadows buildings collapsed into turf, long after the Norse Greenland colonies starved and died in the worsening cold. The C1 mitochondrial DNA continued replicating in Iceland, passed from mother to daughter through generations who had no idea it marked descent from a woman of the indigenous Americas. The molecular evidence persisted when all other traces of the connection had disappeared. Only through the sophisticated techniques of modern genetics could that connection be recovered, and the C1 woman's presence in Icelandic history finally be recognized. What would the C1 woman think if she could know that a millennium after her death, scientists would recover her genetic signature and debate her story? Would she want to be remembered? Would she want her descendants to know their connection to North America? Or would those questions themselves impose modern concerns onto a person whose worldview and values we can barely imagine? We cannot know. We can only acknowledge that she existed, that her life mattered, that her involuntary journey created consequences that rippled through 1,000 years of history. The DNA evidence cannot give her justice for whatever injustices she suffered, but it can ensure she is not forgotten. The story of the Icelandic C1 lineage is ultimately a story about memory and erasure, about what persists and what vanishes, about whose stories get told and whose disappear. For 1,000 years, the C1 woman's story was completely erased, preserved only in molecular form within the cells of her descendants. The genetic revolution has made recovery of such lost histories possible, but it cannot make them complete. We can prove she existed. We cannot recreate her experience. The gap between those two things, between genetic confirmation and historical understanding, remains vast. Science can resurrect genetic ghosts, but it cannot make them speak. The continuing investigation of Norse indigenous contact demonstrates how much remains unknown about even relatively recent history. A millennium seems like a long time, but in historical terms, it is recent, well within the range of detailed archaeological investigation and genetic analysis. Yet fundamental questions about the nature, extent, and consequences of North American exploration remain unanswered. Each new discovery, whether archaeological or genetic, adds detail but also reveals new gaps in our knowledge. The more we learn, the more we recognize how much we do not know, how many stories remain buried in the ground or locked in DNA, waiting for the right techniques and the right questions to bring them back into view. If you enjoyed this video, click on the next one for more lost evidence.